Hi everybody, this is chemistry part 2, so we're going to pick up right where we left off. Let's get started. Our learning objectives today, we are going to work on differentiating between polar and nonpolar covalent bonds. We're going to work on identifying hydrogen bonds, and we are going to work on understanding hydrogen bonds and how they work in actual biological settings. We're going to identify 4 types of biomolecules and try to predict hydrophobic and hydrophilic properties. Okay? All right, now my keys are working. So first we have a little review question. After watching the after watching the part 1 video, I want you to take a look at this question. So true or false, polar molecules have an overall charge, either positive or negative. Is that true or false? Think about that for a minute. Go ahead and answer. And I'm going to point out the important parts here of the question. An overall charge, a polar molecule, does it have a fully overall charge? Not really. So if we think about our water molecule again, right, it has partially negative and partially positive, right? So it's not overall charged. And either positive or negative, well, that's not true either. This polar molecule right here is both. It's positive on one side and negative on the other side. So think about those key parts of this sentence and then decide if it is true or false. Now the next question. So if 2 atoms are sharing electrons and getting rid of their vacancies, they are happy, right? So carbons have 4 vacancies, they can make 4 bonds. Hydrogens have 1 vacancy, they can make 1 bond. Oxygen has 2 vacancies, it can make 2 bonds. So between which of these atoms in cholesterol would you find a covalent bond? So select all of these that apply. This is our molecule cholesterol. We talked about this a little bit in the last one. So first, let's think about what elements are actually here. We have hydrogen, we have oxygen, and we have carbon. Now let's look at our molecule here. Are these elements bonded to each other? Anywhere that a sphere is touching another sphere is a bond, right? Hydrogen is bonded to carbon, right? Hydrogen is also bonded to oxygen. And then we also have carbon bonded to carbon, anywhere these dark gray spheres touch each other. And then we have carbon bonded to oxygen. So looking at all of these bonds, go ahead and make your selection on which of these covalent bonds is present in cholesterol, and we'll move on. So we're going to talk about water. We talked about how water is a polar molecule, and that polarity gives it a lot of special traits. And one very important aspect of water is that water molecules form a lot of hydrogen bonds. Hydrogen bonds are considered a weak bond. Covalent and ionic bonds are considered strong bonds. So a hydrogen bond is when the partially positive side of this water molecule near this hydrogen, it wants to go hang out with the negative side of a different water molecule. So we can see here, this dot line right here, that is a hydrogen bond. Now, the thing is with hydrogen bonds, they do not make water any different, right? So each of these water molecules is still a water molecule. They're just being kind of pulled toward each other, right? So hydrogen can form hydrogen bonds with oxygen, but it can form hydrogen bonds with some other elements as well, as long as they are in polar molecules. So let's think about that. Does hydrogen create molecules? So we just mentioned those two water molecules were being pulled together, but each of them was still a water molecule. Now, if we have a whole lot of water molecules, like we do in this bottle of water, right, all of those water molecules are going to want a hydrogen bond to all the other water molecules. So as those hydrogen bonds are forming between all of our water molecules, is that bottle of water turning into something that's not water? Probably not. So is it creating a new or different molecule? Go ahead and think about that and give your answer. So I mentioned earlier that oxygen is electronegative. Electronegative is like how likely an element is to make a polar covalent bond. So oxygen, if we look here, our scale kind of goes from this yellow color to this red color, and it goes from low to high, yellow to red. And you can see on this subset of the periodic table that yellow is mostly in the bottom left and red is mostly in the top right. So our electronegativity kind of goes up along this blue vector here. So oxygen is fairly high in its electronegativity, is very likely to form a polar bond. Okay, something like sodium, potassium, magnesium, not likely to form polar covalent bonds. Okay, we're going to go out of order on this one. So let's talk about these traits of water. So we have cohesion and adhesion. Cohesion is when water sticks to itself. So this is like when you have water on a quarter. So if we have a little quarter like this, and we have water, let me change colors. Our water is going to be green because I didn't plan this very well. Our water can kind of sit on the top, and you would think that it would fall to the ground once it got past the edge of the quarter, but it doesn't. And that is cohesion, also known as surface tension. Adhesion is when water sticks to something else. So you may have done this in your lab when you have a graduated cylinder. You see a curved edge of water, right? So this curve is because the water is sticking to the side of the graduated cylinder right here. That's adhesion. Water's ability to dissolve substances, we'll talk about that more, but that's part of its ability to form hydrogen bonds. Regulating temperature, if you've ever been near a large lake or an ocean, you may have felt that it's cooler next to large lakes and oceans, and that's because water is sort of a heat sink because it has all of these hydrogen bonds, which means that it needs more energy in order to evaporate by breaking those hydrogen bonds, so it sucks in heat energy. It also expands as it freezes. So some things, when they become solid, they form crystalline structures, like sodium and chlorine form crystalline structures. So all of the chlorines and the sodiums are gonna be like nested, right? They're gonna be really in these like sheets, these very uniform shapes. Water is not like that. Water, let's see, am I over here now? Yes, I am. So water molecules, so we've got our oxygen and our hydrogens. We've got partial positive, partial, nope, backwards. Partial negative, partial positives. Does that oxygen want to be next to another oxygen? No, it doesn't. Oxygen's partially negative. So oxygen only wants to be next to other hydrogens, not other oxygens. So forms that hydrogen bond. You think that hydrogen wants to be next to another hydrogen? No, it wants to be next to oxygen. So it's going to form a hydrogen bond like this. And so because of this, because hydrogen and oxygen are picky, we're not going to get a nice, neat crystalline structure. So the amount of space that it takes for water to make a solid is going to be higher, right? It can't nest as well, so it's going to take up more space. And water is very good in chemical reactions, which is another thing we'll talk about later. Okay. Arrow, there it is. Okay, so let's talk about this for a second. Polar molecules will interact with hydrophilic molecules, ions, and other polar molecules. So if we're thinking about our polar molecule, water, and our ions, sodium chloride, which one of those parts of water is going to want to interact with sodium? So we remember our water molecule. Oxygen is partially negative, hydrogen is partially positive. Which side of this polar molecule wants to hang out with sodium, the positive ion? So go ahead and answer that in iClicker. We're going to talk about biomolecules. Biomolecules are the main molecules that you find in living things, and there are 4 main categories. Now, those 4 categories have subcategories, but you want to remember the main categories first, particularly for the quiz that you have coming up. So our first category is carbohydrates. Now, carbohydrates you may know as things like sugars and starches, the delicious foods like spaghetti and cake. So carbohydrates are ring structures. Their very, very smallest form is a monosaccharide. Mono meaning one. So that means one ring. So ribose is a sugar. It has one ring. Carbon, 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 oxygen. Glucose is a monosaccharide. It has one ring. Carbon, 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 oxygen. And then fructose, monosaccharide, one ring. Carbon, 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 oxygen. Okay? So that is what a monosaccharide is. Carbon ring with an oxygen. Now, disaccharides, di means 2, right? So di means 2, that is 2 rings stuck together. Glucose and fructose stick together, we get sucrose. Sucrose has 2 rings, it's a disaccharide. Polysaccharides over here are long chains. Poly means many, so this is many rings stuck together. Sugar, 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 all the way down. Polysaccharide. Okay. So which 3 elements are making up these rings? You can take a look at this slide, think about it, answer in iClicker. Next, we have lipids, fats. So you might recognize these as oils or butter or waxes, but there's also steroids and hormones in this category. So one of the most memorable is the triglyceride, right? So the triglyceride has this portion up here, the glycerol head of the molecule, and then it has these long tails. And these tails are made up of carbon and hydrogen, and so they are aptly named hydrocarbons. Hydrocarbon tails, right? So if you see a hydrocarbon tail, that's probably a lipid. The other category, steroids and hormones, they have a ring structure, but it is not the same as a carbohydrate ring structure. the carbohydrate ring structure is joined at one place. These are joined at two places, right? And it's not a long chain. It's a small molecule, okay? Well, small is relative. So it has rings, but it's joined in multiple places, and it's not a long chain. Okay, so those are our two types of lipids. So is cholesterol a lipid? So look at this ring structure. Think about it. Think about what we just talked about and answer your question, is cholesterol a lipid? Our third group of biomolecules is proteins. So protein, you may be familiar with this from a nutrition standpoint, things like steaks or fish or pea protein. Those are all nutritionally considered protein. So what a protein actually is a molecule made up of amino acids. So a molecule made of amino acids. Amino acids, you may have also heard, is a nutritional supplement. Amino acids are made up of the same configuration. We've got a central carbon. On one side, we have an amino group. On the other side, we have a carboxyl group. Amino group, carboxyl group, and then an R group. This R group is where we see variation. There are about 20 different R groups, and you may know these as the different amino acids. You may have had to memorize them at some point, so leucine, lysine, tryptophan, things like that. And they typically contain carbon, hydrogen, oxygen, sulfur, nitrogen in some configuration or another. Okay, so proteins are made of amino acids, which are made of these four things: central carbon, amino group, carboxyl group, R group. The big thing about proteins is that they fold. They have several levels of folding. So this first structure is our primary structure. This is the chain of amino acids itself. So we've got central carbon, central carbon, central carbon, central carbon, central carbon, central carbon. So each of those central carbons is an amino acid. That's the primary structure, long chain of amino acids. Secondary structure, what do we see on these amino acids? We see, let me get a different pen color. We see oxygen, right? And we see hydrogen. Now, what do we know about hydrogen and oxygen? Well, if you look at the little drawing underneath me, you remember the type of bond that forms between the oxygens in water and the hydrogens in water. And do you think that those bonds are going to form in other contexts? They are. They are. Oxygen always acts like oxygen. Hydrogen is going to act like hydrogen. So we end up with hydrogen bonds. And those hydrogen bonds produce these helices or these little like zigzags. And that's our secondary structure. Our tertiary structure is when we start seeing a lot of helices and zigzags, right? That's our tertiary structure. Lots of helices and zigzags. Quaternary structure, that's when we have multiple polypeptides, right? So multiple chains of amino acids. So one of those is like heme, which is part of blood, is made up of 4 polypeptides. So that's a molecule, that's a molecule, that's a molecule, that's a molecule, and then they form one big molecule, and that's quaternary structure. Okay? So answer this question in iClicker. Which bond is mostly responsible for the folding, not the chain, the folding? Okay? Now, proteins can be denatured. So if you've ever cooked something made of protein, you know this. So eggs start out as a liquid, and you cook them and they become solid, right? So that is the process of denaturing a protein. So our normal protein in our egg has lots of helices it has lots of those zigzag sheets. Then we heat it up and it denatures. The molecule is the same. All of those amino acids are still here, but they are unfolded. So if they are unfolded, which of those bond types, covalent or hydrogen, is breaking here? Is the protein still, is the egg still egg after you cook it? If yes, then you still have covalent bonds. If your covalent bonds are there, then your egg is still an egg. If your hydrogen bonds break, then you have a cooked egg. It's still an egg, but it looks a little different. Okay? So that's how you can think of that. All right, and our last group is nucleic acids. So this is our DNA and our RNA, right? Our genetic material. So nucleic acids are made up of nucleotides, right? So nucleotides. Carbohydrates are made up of monosaccharides. Proteins are made up of amino acids. Nucleic acids are made up of nucleotides. Okay? So our nucleic acids have a similar repeating structure. So they have a sugar ring, right? So we got carbon, 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 oxygen. That's one part of our nucleotide. Then we have a phosphate group. If you see a P, that's a phosphate group. And then we have the nitrogenous base. And this is where we get variation, right? We're always going to have a sugar. We're always going to have a phosphate group. Our nitrogenous base is where we get the coding part of our DNA. So our nitrogenous bases are adenine, cytosine, guanine, and thymine for DNA. And then in RNA, we end up with a wonky other nucleotide called uracil. Okay, so those are our nitrogenous bases. And when you add those onto our sugar phosphate backbone, you get a nucleotide. And when you stick all of those nucleotides together, you get a nucleic acid. Okay, that is it for our slides today. I hope you learned about bonds.